Hello, OHBM. My name is Valerie Sydnor. I'm a neuroscience PhD student in Ted Satterthwaite's lab at the University of Pennsylvania, and I'm really excited to talk to you today about how the developmental refinement of spontaneous activity varies across a sensorimotor-to-association cortical axis, and how this provides new insight into the spatial and temporal sequence of human neurodevelopment. Human cortical development is a dynamic, protracted, and heterochronous process. During childhood and adolescence, different cortical regions maximally develop and mature at different ages, elucidating the temporal sequence of human neurodevelopment, or in other words, the spatial progression of developmental plasticity across the cortical landscape has profound implications for understanding mechanisms underlying this plasticity in health and disease, age-dependent brain-environment interactions, and the optimal timing for youth interventions. Nonetheless, the precise spatiotemporal sequence of cortical neurodevelopment has yet to be robustly characterized. Extensive prior work has revealed that major developmental changes happen earlier in sensory and motor cortices, shown on this brain in yellow, and later in association cortices, shown in purple. This has been demonstrated with human neuroimaging for large-scale features such as cortical thickness, myelination, and connectivity, and by animal model studies characterizing the maturation of plasticity-regulating features such as parvalbumin interneurons. Critically, beyond this coarse dichotomy, there is additional temporal variability, but a parsimonious framework that accounts for this variability is still being established. In a review article recently published in Neuron, I describe one unifying framework, which posits that during childhood and adolescence, cortical maturation progresses along a sensorimotor-association axis. The sensorimotor-association axis, or the SA axis, is an axis of brain organization that is globally arranged from primary cortices at the sensorimotor pole to modality selective and multimodal cortices in the middle, and then to transmodal cortices at the association end of the axis. This axis captures continuous and concerted variation in remarkably diverse features across the cortical sheet, including but not limited to features like cortical connectivity, cerebral blood flow, gene expression, laminar architecture, and evolutionary expansion. In order to test whether developmental programs may be governed by this SA axis, we studied age-dependent changes in spontaneous cortical activity. Spontaneous or intrinsically generated activity plays vital roles in the developing brain, influencing, for example, neural migration, circuit wiring, sensory maps, synapse refinement, and myelination. Moreover, studies of the developing murine sensory cortex have repeatedly shown that as the cortex transitions from plastic to mature, spontaneous activity evolves from strong and more globally synchronized, as depicted on the left, to sparse and decorrelated, as illustrated on the right. So we aimed to non-invasively study this stereotyped refinement of spontaneous activity using resting state functional MRI. fMRI is certainly not a direct measure of neural activity, but many studies employing simultaneous fMRI and electrophysiology or calcium recordings have demonstrated that a greater level of spontaneous activity or more synchronized activity will increase the amplitude of low frequency bold fluctuations, suggesting that bold fluctuation amplitude may be higher when cortical plasticity is enhanced. We therefore predicted that as plasticity is reduced and the cortex matures during childhood and adolescence, bold fluctuation amplitude would exhibit heterochronous declines along the cortex's SA axis. We tested this prediction in a cross-sectional sample of 1,033 youth ages 8 to 23 years old from the Philadelphia neurodevelopmental cohort. In this sample of participants, we quantified cortical bold fluctuation amplitude by calculating the average power in the time-varying bold signal in a 0.01 to 0.08 Hertz power spectrum. We then flexibly modeled associations between fluctuation amplitude and age within individual cortical regions using generalized additive models, which we use to fit an age spline while controlling for sex and in-scan motion. The first thing that we observed was that the magnitude and direction of bold fluctuation amplitude age effects varied across the cortex. We further visualized this variability by plotting fluctuation amplitude age fits in individual cortical regions. So each line here is a region, which revealed this cortical continuum of developmental trajectories. And in general, sensory regions, for example, primary visual cortex shown here, exhibited large and earlier declines in fluctuation amplitude, which are potentially indicative of a decrease in or decorrelation of spontaneous activity. Higher order association cortices like the DLPFC, on the other hand, tended to show inverted U-shaped curves suggestive of heightened synchronized activity in early adolescence. We next tested whether developmental change in bold fluctuation amplitude was related to the maturation of intracortical myelination, which is a very well-established plasticity limiting factor. We compared our data to results from Graham Baum and Leah Somerville's recent paper characterizing the development of the T1-T2 ratio in youth 8 to 21 years old. Fluctuation amplitude and T1-T2 ratio overall age effects were very strongly inversely correlated, indicating that cortical regions that undergo larger increases in myelin content from childhood to adulthood additionally experience larger decreases in bold fluctuation amplitude. In addition, the age at which fluctuation amplitude began significantly decreasing within each region was associated with the age at which the T1-T2 ratio maximally increased. This association reveals temporal correspondence between fluctuation amplitude development and maturation of a main limiter of cortical plasticity. Having established that fluctuation amplitude development tightly paralleled development of a plasticity regulator and broadly diverged between sensorimotor and association cortices, we sought to determine if developmental patterns spatially conformed to the SA axis. To accomplish this, I ran a PCA on regional fluctuation amplitude maturational trajectories. The first principal component shown here explained 87% of the variance in development of this functional measure and can therefore be conceptualized as the principal axis of spontaneous activity development. This principal developmental axis showed striking homology to the SA axis, with principal developmental axis loadings and SA axis ranks correlating at R equals 0.7. Further, if we divide the SA axis into 10 percentile bins and average regional age fits within each bin, we can see how the continuous spectrum of developmental trajectories visible at the regional level is recapitulated by SA axis percentile bins. Finally, we wanted to elucidate whether this sensorimotor-to-association neurodevelopmental pattern dominated at every age between 8 and 23 years of age, or if it was most pronounced in a specific age range. To explore this, we quantified age-specific developmental effects at approximately 1 month intervals between 8 and 23. So here, for example, are fluctuation amplitude developmental effects at ages 10, 15, and 20. We then correlated age-specific developmental effects with SA axis rank. This approach generates age-specific correlation values that quantify the extent to which fluctuation amplitude maturational change is spatially ordered along the SA axis at a given developmental time point. If we plot age on the x-axis and age-specific correlation values on the y, we can see that there is a robust correlation between developmental change and SA axis rank from 8 to 17, with peak alignment to the sensorimotor-association axis observed at 15 years of age. Interestingly, this correlation rapidly dropped to 0 near the end of adolescence, suggesting there may be a program switch in the spatial patterning of developmental processes after age 18. Altogether, this work provides evidence that neurodevelopmental processes unfold along a sensorimotor-association axis of cortical organization from childhood to adolescence. Our findings linking a main developmental axis to a prominent spatial feature axis also indicate that temporal developmental variability may produce spatial feature variability, providing insight into developmental origins of adult brain organization. Finally, our data suggests that during adolescence, spontaneous cortical activity increases in strength or coherence in transmodal association cortices, which is a functional hallmark of developing plastic cortices. This provides support for the idea that early adolescence may be a sensitive period of brain development characterized by heightened malleability in the association cortex, and thus that educational, social, and psychiatric interventions may have a large impact on higher order areas of the brain in late development. I would like to conclude by giving major thanks to the entire Penn Lifespan Informatics and Neuroimaging Center, especially to our unstoppable informatics team, to our collaborators on the study, and last to Bart Larson, who is essentially my scientific advisor, statistical teacher, and academic role model, and to Ted Satterthwaite, who doubles as my PI and my biggest supporter, and who is without a doubt the best graduate school mentor that I could have. Thank you, OHBM.